Welcome to our second lecture. Today we will be exploring how gender is determined by genes and by hormones in the human body. To the right of the screen you can see a photo of 23 pairs of chromosomes, 22 of which are autosomes and the last pair are sex chromosomes. In this case there is an X and a Y chromosome present, which makes this a male from a gene standpoint. We will talk about this in a little more detail shortly. Both males and females have three sets of structures: the gonads, internal genitalia, and external genitalia. These gonads produce gametes, which are the egg or the sperm. In males, the testes are the gonads that secrete sperm, and in females, the ovaries are the gonads that produce eggs. Germ cells are the undifferentiated gonadal cells that will either produce eggs or sperm. The internal genitalia are made up of the accessory glands and ducts that connect the gonads with the external environment. And finally, the external genitalia are the external reproductive structures. So here we can see on the right an image of male and female structures where you can see the testes are the gonads of the male system and the ovaries for the female. Every cell of the human body that contains a nucleus has 46 chromosomes. Of these 46 chromosomes, there are 22 pairs of autosomes, which what that means is that there are 22 matched pairs of chromosomes. And then there is one pair of sex chromosomes, so that makes a total of 46. There's also this is also called a diploid number because the number of chromosomes are in pairs, so the amount of DNA in a diploid cell can be expressed as 2n. The 22 autosomal pairs will guide our cells to develop our human body and other specific characteristics of our body. But this is the case for every cell except egg and sperm cells. We'll get to those in just a moment. So the sex chromosomes are either an X or a Y chromosome. So these will direct the development of both the internal and external genitalia. And the X chromosome is a larger of the two, which contains many of the genes that are missing from the Y chromosome. Eggs and sperm have 23 chromosomes, and these chromosomes have 22 pairs of autosomes and one of the sex chromosomes, either an X or an Y. Or I should correct and say that these have one chromosome from each of the 22 pairs of autosomes, and then one sex chromosome, either X or Y. So these are haploid cells. So the amount of DNA in these cells can be expressed as 1n. So when an egg and sperm come together, they create a diploid, and the zygote has a set of 46 chromosomes with 22 matched pairs and one pair of sex chromosomes. The pair of sex chromosomes contains one sex chromosome from the mother and one from the father. So here on the right we can see the 22 pairs of autosomes, and then the X and Y chromosomes. So this is a diploid right here. Alright, so we'll move on to genetic sex and sexual differentiation. So genetically, females who inherit one X chromosome from the mother and one X chromosome from the father are genetically female. Males who inherit one Y chromosome from the father and one X chromosome from the mother are genetically male. Without the Y chromosome, there will not be development of male reproductive organs. There are cases where, in the event that there are multiple sex chromosomes in a zygote, for example, if there are two X's and one Y, like XXY, the zygote will still become a male. However, a zygote that only inherits one Y chromosome and nothing else, the zygote will die because the larger X chromosome is needed for necessary function, and it contains all the necessary genes for that. In the event that only an X chromosome is inherited, the zygote will in fact develop into a female, but the two X chromosomes are needed for complete normal function. In a female fetus, once ovaries develop, one of the two X chromosomes is inactivated, and this becomes what's called a Barr body, which is just a clump of nuclear chromatin, and it eventually degrades. Oops. The bipotential gonad is what the embryonic tissues are called before they have differentiated into male or female tissues. This consists of an outer cortex, which in the absence of SRY gene will develop into ovaries, and in the presence of SRY gene, which is the sex determination region of the Y chromosome gene, will develop into testes. The bipotential gonad also has external genitalia, which consist of the genital tubercle, urethral folds, urethral groove, and labioscrotal swellings. The internal genitalia consist of Wolffian ducts, and Mullerian ducts. So on the right here we can see the bipotential gonad separating into male and female components, and we'll continue our discussion on the next slide. The SRY gene produces something called the testes determining factor, TDF, which then will bind to DNA and then activate other genes that produce proteins to direct the development of the medulla into testes. Testes development does not require the androgen testosterone, but after testes differentiate, they will secrete anti-Mullerian hormone, AMH, from Sertoli cells, and they will also secrete androgens, testosterone, and the more potent version DHT, which is produced when 5-alpha reductase converts testosterone into DHT, and those are secreted from the interstitial cells, also Leydig cells. They're also called Leydig cells. And testosterone and DHT are the dominant steroid hormones in males. AMH causes Mullerian ducts to regress, while testosterone causes Wolffian ducts to become male accessory structures like the epididymis, vas deferens, and seminal vesicle. Testosterone will control the testes lowering from the abdomen into the scrotum. DHT controls the rest of male sex characteristics as well as the external genitalia. In the absence of SRY gene in the female embryo, the cortex will develop into ovarian tissue. There are many genes required for development of functional ovaries. Also, in the absence of AMH, the Mullerian ducts will become the upper portion of the vagina, uterus, and fallopian tubes. In the absence of testosterone, the Wolffian ducts will go away, and without DHT, the external genitalia will start to take on female characteristics. So to wrap things up, there are many things that go into determining someone's sex. There are genetic, chromosomal, gonadal, morphological, and psychological components and characteristics. For one example, an individual could be an XY, have the XY sex chromosomes, but could appear to be non-male due to development issues somewhere along the lines. So, and also separately, it's been gaining ground for people to be able to choose their sex when they're old enough to do so, and what this is called when what a person considers him or herself to be is called their gender identity. So this wraps up our discussion of our determination of sex from genes and hormones. So thank you for listening.